Today we're going to focus on proteomics datasets in VEuPathDB sites. And the goal is really to walk you through a number of the searches that we make available to you based on the proteomics data that we load. And we'll touch on three types of searches. We'll touch on searches that interrogate peptides that come from mass spec experiments. We'll also touch on searches that interrogate quantitative proteomics data. So we have different types of searches that allow you to look for proteins that are changing in expression levels based on their proteomic experiments. And also we will look at data that we have that interrogates the post-translational modifications, so PTMs. So if you're interested in proteins that are potentially phosphorylated or acetylated, we will walk you through those searches. And throughout this, as usual in our webinars, we'll try and integrate other searches or other features just so you get a feeling of what else you can do with these searches and the results. So quickly, just to give you a little bit of background, and I'm not going to go into too much depth in all of this just because of the time limitations, but just so we're all on the same page, you can assay proteins in different ways, but typically, traditionally you used to run this on gels, obviously, and you get all these spots, but these spots are actually proteins or fragments of proteins, and their chemistry basically tells you something about what they are. And of course, methods, high throughput methods rely essentially on the same notion, is the chemistry of the peptides, to be able to then identify what those peptides are and the quantity of those peptides. And so there are many different methods, but ultimately you will take proteins and you're gonna break them up somehow or label them somehow, depending on what you're doing with it, whether it's quantitative or if you're just trying to determine the peptides that are in your sample. You typically do some kind of chromatography on your peptides, and then in order to identify these peptides, you have to somehow detect them and detect their size, basically, their mass. And so to do this, you have to charge these molecules in a way. So you ionize them, and then you have to have some kind of detector to detect these peptides. This is just a quick schematic. So you have your complex protein mixture. You break them up into peptides, usually using an enzyme like trypsin. And we know where trypsin digests. So that comes in very handy in your computational methods that are part of your mass spec identification method, which we'll talk about briefly later on. You ionize the peptides, and again, you go through maybe a chromatography, and then you do some kind of detection. And this is just a quick schematic to show you what this detector may look like. So you have an electron beam, which will ionize your peptides, which came in here. These peptides then are going to go through this accelerator, and they're these essentially really big magnets, and they're gonna, because of your peptides, they're gonna ionize, they're gonna have different charges because of their different composition, basically. They will slightly move away from the central line, basically, and they will then be detected at different spots on this detector. Now, based on this angle and where they fell on the spot, basically, you can determine what the mass is of a particular peptide. So that's pretty cool, and it's quite effective. But then ultimately, what you do is you have your results from your mass spec experiment, but now you have to determine, well, I have all these masses, right? I have this file full of masses, and I need to figure out, well, what do these masses correspond to? Which peptides do they correspond to? And so since you're doing an experiment on a known organism, so in theory or typically, so let's say you took toxoplasma and you were doing a proteomics experiment on it. So you purify the parasites, you do the whole experiment, so now you have these masses, and you want to figure out, well, what did it belong to? So what you do is at the same time, you do an in silico theoretical, essentially mass spec experiment on your predicted proteins from the genome, right? And since we know where trypsin digests, you can take a file of predicted proteins, electronically digested with trypsin. Now you have all these digital peptides, which now you can, again, computationally determine what their predicted mass would be. and then you basically correlate them. And you say, oh, well, this one is actually this one here. So this is the peptide that comes from a specific protein, right? Now you can match it to that protein with a certain level of confidence. We do get, I'm going to quickly switch back to Firefox here, and I'm just going to show you ToxoDB as an example. We do get the question often, well, I'm running this experiment. I need to know where the file is that I'm going to use to generate this electronic signatures, electronic mass spec, basically, for my analysis. And so if you go to any of our resources, any of our databases, you'll notice that we have a section here called data. And there's one called download data files. And this allows you to download the large data files, whether it's the whole genome or whatever. You'll see here there are files for every single one of our releases, going back to release 10, I believe. That's the earliest one we have. You can always click on the current release. And now you can scroll down here. You can do a quick find on the page if you want. But depending on what you're interested in, you may, typically people in Toxoplasma will be interested in ME49. So I can select this, I can go to my FASTA files, the data, and you'll see here now I have different types of FASTA files, which is essentially a structured text file. And in this case, for my experiment, I'm interested in the protein file, the protein FASTA file. So this is the one you would use for your proteomics analysis. And so you can click on this and download it, and see here it's 9 megabytes in size. Okay, so let's keep going. Okay, and that's basically now you can map the signature, this peptide signature, back to the protein, and that's how you can determine what proteins showed up in your particular experiment. Okay, for quantification, again, there are many different labeling methods and schema, and you'll see in different publications they have different ways of labeling. There's a label-free method, and that often looks at the number of spectra that come out in your particular experiment. And so for each peptide, it may show up multiple times in your experiment, and the more it shows up in your analysis in that detector, basically in the beam I showed you earlier, that gives you kind of an indication that, oh, that's occurring more than other things, right? So if something shows up 10 times and another peptide shows up 1 time, right? The one that shows up 10 times is likely to be more common or more quantitatively more in your sample. And then there are other types of methods here, which I won't go into detail, but suffice it to say you're typically doing some kind of a comparison between a labeled and unlabeled peptides, and you're trying to determine what is the relative quantification or quantity of your peptides that you're interested in detecting. Okay, so this is it in terms of intro. So what we do in the database, in any of our databases, we take either the peptides that were identified in an experiment, and we map them back to the translated proteins and also back to the genome. So we'll get the location of a peptide not only on the protein but also on the genome, so we can get the coordinates for that. We do not. The one thing we do not do is we do not take your original mass spec file, and then we do not do a research of the database, right? So technically, in a perfect world, you would want to do that, right? But that's incredibly compute intensive, and that ended up being too challenging to do on a regular basis. Compared to RNA sequence data, we reanalyze all the RNA sequence data, and that's more streamlined and works well. But with peptide data, that proved to be complicated. So what we decided to do is we simply take the peptides from the experiment that were identified, and we map them 1-to-1. So if a peptide has a particular sequence, and that sequence is found in protein A, then that peptide goes to protein A, basically. For quantitative data, we also take the quantification that was developed in the experiment, and we load them. So each protein will get a particular value, and depending on the experiment, it may be a direct comparison. So then sometimes it's not a value, but you're really just comparing fold differences between conditions. And if you go, if you want to search for proteomics data in any of our resources, you'll find here on the left we have these specialized searches, and they're categorized in different ways. But and of course, these categorizations may be useful to you or maybe not. Sometimes they're easy to interpret, sometimes they're not. So one of the easiest things to do is to just filter your searches up here by just starting to type the word you're interested in, right? So for example, I'm just typing prot and trying to see what comes up. And so you keep typing and the searches keep filtering. You notice here now the category called proteomics. And here are 3 searches that are available to you. You can search for mass spec evidence, and this is looking for peptides. And Suzanne is going to demonstrate this in a second. You can search for post-translational modifications. So I can say, give me all proteins that have a phosphorylation site, at least 1 or 2 or whatever. You can change that. And finally, I can search for proteins or genes in this case as a surrogate for proteins based on quantification. And of course, depends on really the experiment that you're looking at. So we will demo all of these types of searches for you right now. And so I will pause for a second as we switch over to Suzanne. If you have any questions, feel free to put them in the Q&A section. And Suzanne, I'm going to go ahead and make you the presenter right now. Let's see.
Okay.
And you should have gotten a note, I think, now.
Oh, yeah.
Okay, perfect.
Okay. So as Omar said, I'm going to demonstrate the peptide searches. And these searches return genes based on mapped peptides from proteomics experiments that we've integrated into our databases. They give evidence of expression at the protein level, right? So I'm going to demonstrate one in Giardia, and so I'll go to this to the search page. There's a couple different ways to do it. This search for menu, or we have this menu up at the top here too, which is available from every page on our site. So I'll open genes, proteomics, and we have 2 searches available here. We obviously don't have post-translational modification data in Giardia, so that search is not offered here. So I'll do the peptide search. It's called mass spec evidence. And so I come to the search page and I have a list of parameters that most have default settings, the organism or experiment, often you have to choose, but I have parameters that need to be completed before we run the search. I have the get answer button when I want to initiate the search. And then scrolling down further, I have a description of the search, which will help you understand what the parameters do and what's actually returned. And then down below that, I have a list of data sets that are used in this search. And these link to our dataset records. So if I'm going to concentrate on this one, the Giardia secretome from intestinal epithelial cells. So if I click on this, then I will go to a record page that describes that experiment. And there's lots of information, including the authors, when we integrated it, and then links to searches and other stuff in our site. So let's concentrate on this experiment, this data set. And I have a little, let's go back up. But I also have a little, yeah. So what these guys did, and they published in Neglected Tropical Diseases, they identified major excretory or secretory products, and they called it a secretome, when Giardia trophozoites were incubated with intestinal epithelial cells. So they, did a little bit of a time course. They incubated for 12 and 6 hours and collected samples and then did their proteomics. But they also did controls where they incubated Giardia without the intestinal epithelial cells, so axenic cultures as control incubations. And they did this in 2 Giardia strains, the WB and the GS. So we have that data now integrated, and this is the search. So I will, we need to now choose an experiment to search. So I'm going to go ahead and expand all and look for the IEC. And we see both of the experiments here, assemblage A, WB, and assemblage B, GS. And I'm going to go ahead and choose the secretome in assemblage A and choose the non-control samples. So I'm going to look for genes with mapped peptides in that were present in cultures that were incubated with intestinal epithelial cells. And then we have two other parameters here: minimum number of unique peptide sequences and the apply the number of peptide sequences on a per or cross sample basis. The minimum number of unique peptide sequence defines the number of peptides that a gene needs to have mapped to it to be returned by this search. And the lowest number, of course, is 1. But you can, if you require more peptides to be assigned to that gene, then you're making the search more stringent. So, I mean, we could leave it at 1. Let's go ahead and put 2. And then apply the minimum number of peptide sequences on a per sample or a cross sample basis. So the default is per sample, but what that means is that we're asking that at least 2 peptides be assigned, be mapped back to genes in order to reduce for it to be returned by the search. But the thing is, we could, if we choose across samples, one of those peptides could originate from this sample, and one of those peptides could originate from this sample. If we leave it at per sample, then we're saying that before a gene is returned by the search, it needs to have 2 peptides from this experiment, from this sample, to be qualified to be returned. Okay, so I have set up the search. I have 2 samples that I'm searching. I want each to have 2 peptides mapped to it, and I'm going to click Get Answer. And I am returned 248 genes that have that were expressed in these samples. So let's just go ahead and look at the results. Here on the left, we have the organism filter, and I can expand all of that. And I see that, all of my hits are in Giardia assemblage A, and that makes sense because that is the type of data that we were searching. And then I have a table of results. These are the first 5 of gene IDs that were returned by the search of the list of 248, and then associated data. And there are 3 columns that are unique to our peptide searches. This first one here is the selected samples that match. So each sample matched the qualifications of this, the criteria of our search, and identified this gene to be returned by the search. There were 73 unique peptides associated, and 42 across all samples, right? So, and then I can also, to dig a little bit deeper, I can go to a gene page and look at peptide evidence or proteomics evidence there. So let me just show you the proteomics section. I'll go down here, use this navigation tool to go to proteomics. And basically, there's 3 sections of data that we give in the proteomics section. The first is a table, and now this is all associated with this gene. So for each of these samples, these are the number of sequences that are supported by this number of spectra for this gene. And then we also have a graphic table that is nice to look at. And so along this axis here, little error, those things happen. So along this x-axis is the protein, and it's actually kind of long, 2,000 and so amino acids. And then each of these is a different experiment. And the glyphs within the experiment represent the peptides that are mapped to that gene. And I can click on this peptide and get more information. It was from this experiment, it was from the exenic 6-hour time point, and this is the sequence that mapped back to the gene. Okay, and then we have one more thing, the quantitative mass spec data. And these come to us really as a table of numbers, right, of expression values. Oh, here we go. Sorry about that. Come to us as a table of expression values, which is actually available here if you want to open that up. But then we graph it and show you, its expression, this gene's expression in each of these samples. Okay, let's go back to our gene search. So what was great about this experiment is that they actually had controls, right? And so in this, our first search, we only searched the experimental samples, right? Those that were incubated with IEC cells. So it'd be nice to find out what genes are unique to those samples, so we can subtract out the controls, and we can do that with our search strategy system. So I'm going to add a step and choose to subtract a new search that I run on the controls. So I'm in my AdStep pop-up. I'm at the Proteomics search. I'm going to choose assemblage A, 2 controls from that experiment. I'll go ahead and set my minimum number to 2 just to make it even Steven. And that search, so the controls contained 196 genes, whereas the actual samples contain 248 genes, and that leaves us with 87 genes that are unique to those samples that were incubated with intestinal epithelial cells. So that is how to dig deep into that experiment. If there's no questions, then I'll move on to another example.
Yeah, so far no questions, Suzanne. So I think it's crystal clear. So keep going. And again, people ask questions in the question panel if you have them.
Okay. So the next thing I'd like to demonstrate is in pure plasma DB, and I'm going to pretend I'm interested in Schizont proteins, and you don't really have to pretend about that, I guess. But so I'm going to ask the question, what genes are expressed at the protein level in Schizonts? And I will go to my mass spec evidence search and look for an experiment to see if I have that. So I'll use the search for Schizont, and I find that we actually have quite a few Schizont samples, and they're from 2 different experiments. And you know what? That's okay. I'm gonna choose the total proteome, Schizont proteome here, and the Schizont total proteome here, and go ahead and look for and run this search. Leave it very broad on a per sample basis, and click Get Answer. And what I come up with is 1,493 genes that are expressed in the Schizont, and that is evidence that comes from 2 different publications, right? So this is just an example of using 2 different experiments to support, to find evidence to support your answer or question of what genes are expressed at the protein level in Schizonts. So that's a quick little demonstration there. Let's go on to PlasmoDB. And here, I'm going to ask the question, what genes are expressed at the protein level in gametocytes, right? So I'll go to the mass spec evidence search. And the first thing I notice here is that that there are actually 4 data from 4 organisms represented. And if I expand all, there's a lot more data in this available for the Plasmodium species here. So let's go ahead and look for gametocyte samples. and I find 1 experiment in P. burgii and then 3 experiments in P. falciparum 3D7. So, you know, I'm just going to go ahead and click them all. Let's look to, I want to find genes that are gametocyte expressed. Might as well search everything. And what I find is we have about 5,400 genes returned. And if I look down here, I see that about 2,000 of them are from P. burgii, and 3,400 of them are from P. falciparum 3D7. If I could open that, there we go. Yeah. This is really neat, and you can, look further into each of these proteins, possibly add some steps to see some other biological properties that are shared by these genes. But if I'm a P. falciparum 3D7 researcher, I'm not real familiar with these P. burgii genes a lot of times. So I would like to show you a way to transform the P. burgii genes into P. falciparum 3D7 to get a little bit more comprehensive look at the falciparum genes, right? So I'm going to go back here and edit this search to remove that sample that we added. So I clicked edit up here, and now I'm going to revise, hopefully, and expand all. And this is my P. burgii ANCA. I'm going to uncheck that. Let's not search that this time. The only other ones that I'm going to search are the falciparum 3D7 gametocyte. And in fact, that's kind of a mistake. We just want to do this one sample, okay? Because I had actually checked trophozoite, sporozoite, and merozoite also. But now we have gametocytes stage 4 and 5, gametocytes here, and male and female gametocytes, all in P. falciparum 3D7. I'll click revise. And this search returns now 2,864 genes that are all in P. falciparum 3D7. But we know there's more evidence to be interrogated here, right? That P. burgii experiment. So I'm going to click AdStep and say union my first experiment with a new search for peptide-based data in P. burgii. And I'll choose male and female gametocytes and run that search. Right, and now I have 4,800 genes that are spread across P. burgii and P. falciparum. But here's the little trick. I can transform these P. burgii genes into P. falciparum by using our transform by orthology tool. So I'll click edit. Nope, no, I won't. I will click, yes, I will, sorry. I will click edit, make a nested strategy. Now I can perform actions on this, these results before they get added into our P. falciparum. I'm going to click Add Step. Transform by orthology into orthologs of P. burgii. Run step.
Falciparum, actually, you made burgii.
Oh my goodness, I'm sorry. I really need a coffee.
I think as you're doing that, one cool thing, this orthology step, it allows you to really leverage experiments from across species, as Suzanne was indicating. And so she's doing a union here because she's saying, I want to cast the widest net. I want to find the largest number of potential gametocyte genes that are expressed from proteomics experiments. But you could imagine also doing this, and maybe Suzanne will demonstrate that in a second, is changing that to an intersect if you want to simply say, well, if it's in gametocytes in falciparum, and also in gametocytes in falciparum orthologs of Burgii ones, then I'm interested in those. Those will give me higher confidence, for example.
Yes. Yeah. Yeah, that's neat. Right. So these are our P. Burgii genes. There are about 1,900 of them. That's associated with about 1,800 P. falciparum orthologs. That gets shuffled back up into here. And when I add that to this P. falciparum other results, I come up with 32, almost 3,300 genes. And as Omar says, I might want to find genes that are in common between these two. I can edit this, change it to intersect. And I have about 1,300 genes that were present in gametocytes in P. Burgii and present in gametocytes in P. falciparum 3D7. And this is expressed now within our system as P. falciparum 3D7 genes. Does that sound good?
Yep, it's great. Were you going to demo anything else, or should I take over with the quant?
I think that's it. I was looking at the questions. Looks like they've all been answered. So
yeah, cool. Okay, so let me switch back and make myself the presenter. And I'm going to try and answer a question that came in. So the question was about analyzing one's own mass spec data and then comparing it to data in the database. So one thing, unfortunately, right now you can't do. We don't have a pipeline where you can take your raw mass spec data, your peak files usually called, and then do a research of the proteomics database to find mass spec hits. That's a bit too compute intensive to run on just sort of on the fly like this. There are mechanisms out there and something potentially worth investigating in the future, but for now that's not possible. But you could imagine a scenario where you have a list of genes that came from an experiment you did in the lab, that from a mass spec experiment, right? And so you did your experiment and you did all your analysis, and you're like, oh, look, I have whatever 100 genes, and here I've taken a list of whatever 67 random genes from Cryptococcus in this case, and so I can take these genes and I want to ask, well, I want to put them into FungiDB in this case. These are fungal genes, because I want to ask additional questions about these genes. So I can go back here to the database. I'm going to go to FungiDB, and the first thing I want to do is I want to take this list of genes and put them into a search, right? So I can actually do other things with them. And so these are lists of gene IDs. So I'm going to look for a search for IDs, right? So I'm just going to, in my left-hand panel here, I'm going to type the word ID, and looks like I can find genes based on gene IDs. So let's go ahead and try this. So I'm going to edit this step. I'm going to paste my IDs and get answer. And so now this will give me a step in a new strategy, right? This is my new experiment with my 67 genes that I found really interesting, right? And I'm going to then do something else with them. So now I can ask, I wonder if any of these 67 genes were also detected in an experiment that was available in the database. So let me go ahead and add a step here. And since we're doing proteomics today, and Suzanne already demonstrated the peptide searches, I'm going to demonstrate the quantitative searches. And so let's go ahead in this. So I'm adding a step for new genes. I'm going to intersect my 67 genes with a new search. And the new search, again, I'm going to look for proteomics, and it looks like there's one for quantitative data. So I'm going to click on quantitative data. And now I'm going to look and see, well, what is available to us. There are a couple of Cryptococcus experiments right here. So let's go ahead and one of these. So I'm going to run this one that's called a direct experiment. So the DC here. And if you look at the top here, there's a little description of what this is. But also when you go to the search, so if you click on DC on this little button, you'll get the search parameter. So this is a direct comparison. And of course, you're looking at this and like, well, I wonder what this experiment is all about. And as Suzanne demonstrated earlier, for each experiment, we have a data set page. So it gets its own data set page where we try and describe it as much as possible. And sometimes we need more information. So if you ever come across a data set page and you have more information to contribute to it, let us know and we'll go ahead and fix it. But if you click on this, so I can click on this, I open this up already in a separate page. you'll get more information about this particular experiment, obviously including a link to the publication and some more information. And quickly here, I could tell that this is an experiment where they incubated Cryptococcus neoformans with either in glucose containing conditions or galactose containing conditions, and that's to repress pKa1 or to induce it. So under galactose, it gets induced. And so back to my question, I'm asking, well, I wonder if my 67 genes are also expressed in, let's say, galactose, under galactose or pKa, I think, inducing conditions. And I can change this to either upregulated, if you want to look for things that are specifically upregulated, or I can leave it at up or down regulated. You don't care, as long as it's differentially regulated in this experiment, then I would like to know if any of my 67 genes were also present there. And so you can run this search. Let's see what happens. So it tells me that there were 1,469 genes that were differentially regulated in that experiment, and 29 of them were in common with my 67 genes that I uploaded, right? So this is a one way where you can interrogate your own lists of genes that came from an experiment or your favorite genes or whatever. You have a list of genes or from a publication, right? And so you put them in as a gene ID search, and then you can run a separate quantitative search. So I'm going to jump to TriTripDB, and I'm going to demo another quantitative search. So again, right here on the left, I'm going to just filter the results. I can either do proteomics, or now I know I'm going to look for quantitative, so I can directly find the quantitative mass spec evidence experiment. You'll notice here a ton of experiments. And so I could come in here as a researcher saying, well, I know there is an experiment in T. brucei for cell cycle proteomics. I just can't remember the details of it. So I'm just going to type in this filter cycle, and now at least you have a limited number of experiments to look at. And now I can see that the first one here says T. brucei procyclic cell cycle proteome, and I think, oh, this is maybe the one that I want to search. So I'm going to search it, and you'll notice that it has a fold change search. It's not a direct comparison. So I'm going to go ahead and click on fold change, and I'll walk you through this experiment. So this is the same experiment or same search that we have available for any fold change data, including RNA sequence data. You'll get the same type of mechanism. And the idea here is I'm going to look for genes that are, let's just decide what we're going to do. We'll look for genes. So remember, this is a cell cycle experiment going from half an hour all the way up to 11 hours in the procyclic cycle. So these were synchronized cells, basically. And so now let's say I'm looking for things that are upregulated at the end, towards the end of the cell cycle, and compared to the beginning of the cell cycle. So I'm going to pick a couple of time points at the end of my cell cycle. Let's pick 3, and I'm going to pick 3 at the beginning of my cell cycle. Okay. And you notice here there's a graphic on the right, that gives you an indication of the type of gene that's going to be returned based on the parameters you've selected on the left. And so in my case, I really want to find things that are really going up at the end compared to the bottom. Here, this here won't really give me the results that I want. You can see the samples are kind of just all over the place with the fold change. So I'm going to change this to, and there are different options. I can go average, right? And so this means the average between the 3 red time points right here is going to be 1.5-fold higher than the 3 blue samples right here, okay? The 3 blue time points. Or I can be a bit more stringent and say, well, I want to compare the maximum to the minimum here. So this means that the minimum expression value of my 3 red time points later in the cell cycle are going to be 1.5-fold higher than the first 3 time points, okay? So this is the more stringent one. So once you set it up and you're happy with what it looks like, you can go ahead and get answer. All right, and so now this tells me that there are 4 genes that met this criteria. Okay, and now you can go down and look at them. And it's important with any database you're looking at to actually, you're the scientist, right? And so you have to make sure you're looking at the results and saying, well, do they make sense? If you're getting results that don't make sense at all, it doesn't mean either you did something wrong in setting up the question, right? And you can go back and check it, or maybe there's something wrong in the database. So always feel free to click on contact us and let us know if you're having problems or concerns or not sure about something. So here are my 4 genes of interest, and you can look at the graph here, which shows you all the time points, and you can see, oh yeah, this meets the criteria, right? The latter time points are higher than the earlier time points, so that's good, right? These are samples that are genes that are upregulated based on quantitative proteomics later in the cell cycle, in the procyclic cell cycle, compared to earlier in the cell cycle. And you can look at them and you'll see things, hopefully things here look interesting. Aurora B kinase, I think that makes sense, involved in cell division, so that's probably makes sense. Okay, so let me pause for a second and see if there are any questions in the question panel. I don't see any, I think. Okay, good. So we'll continue. So the next thing I'm going to demo is, I think I will jump to ToxoDB. And again, I'm going to pick the proteomic quantitative proteome mass spec experiment, and let's take a look here. And what we can do is, again, you can pick any of these experiments, and you'll see here there's fold change, there's DCC, direct confidence comparison. So direct confidence, let's take a look at this one. Actually, I haven't run this search yet today, so let's give it a try. So DCCC. So this means that it's going to be a direct comparison, but with some confidence as well, right? So I can pick now these samples that I'm comparing to each other, and sometimes they're different samples, obviously, depending on what the experiment was. I can look for up or down regulated or both. But now, not only can I change the fold change, but I can also play around with the p-value, because this particular experiment came with some confidence values for each of the expressions. Typically, I would run this as a default and see what comes up, and then you can always modify these searches and see if you get anything interesting. So in this case, I got 163 genes that met the criteria. Again, each gene will have a graph. These graphs are also represented on the gene pages, as Suzanne showed. But I can edit any of these steps, and I can revise them and say, well, twofold maybe was too low. Maybe I want to look for things that are fourfold, so I can go down and change this to 4. Or I can change the p-value, obviously, and then see what happens. At some point, you'll get so stringent that you won't get any results back, that's okay too, as long as you know that you can edit the step and revise it. So I'm going to jump to PlasmoDB here, and I'm going to highlight a couple of searches. So I'm going to do one more quantitative search, but then I'm going to transition to the post-translational modification searches, which are also based on the proteomics experiments. And so let me go ahead and do this. Let's do quantitative. So here's my quantitative mass spec experiment. And there's an experiment in here that looks for genes that are based on the proteomic, experiment, quantitative proteomic experiment, that are quantitatively found in the apicoplast, right? So this is this plastid-like organelle in apicoplexan parasites, like malaria and toxoplasma. So now I can go in here and say, well, I want to find genes that are up in the apicoplast compared to the ER. I can change this to upregulated. I can even change the fold change. Let's change it right here to 2, right? So I'm looking for genes that have a 2-fold higher level of protein in the apicoplast compared to the ER in this case. And you can get answer. And so this tells me that there are 225 genes that met this criteria. So I do know that there's an algorithm in PlasmoDB for apicoplast targeting. So I can add a step here, as Suzanne was showing how you can integrate different types of data. So I can add a step here and say, well, I want to look for subcellular localization. Let's look for localization. There is Plasmodium falciparum subcellular localization. And it's a very simple search. It just says, is it localized in the apicoplast, right? And I'm going to intersect, because I want to look for things that are both in my proteomics experiment and in this computational method for detecting genes that are, or proteins that target to the apicoplast. And I'm going to run this search. And so now you will see that I get 138 genes that met these criteria. Okay, so this could be potentially my list of high quality, definitely going to the apicoplast genes that I'm interested in. So I can take this a step further, and remember that for depending on the mass spec experiment, you can also detect post-translational modification events. And so, and if we have that data, obviously we load it and provide you with searches. So I'm going to add this a step here, and again, I'm going to look for proteomics experiments. And you'll notice here one of the options is post-translational modification. So I'm going to select that. You'll come up with this type of PTM basically, and you'll notice they're different depending on the on the experiments we have, we'll load them. But in this case, I'm interested in phosphorylation sites. And so let's keep the rest of the defaults, but I'm basically asking how many of the genes that are expressed in this ER apical proteomic experiment that are also found in the computational method for apical targeting are phosphorylated. I don't know what the answer is, so this is a real experiment, so let's find out. And it turns out 16 of my genes are potentially phosphorylated and are localized to the apicoplast, right? And then you can go down and look at them. And any of these steps can be edited at any moment, right? So I can go ahead and edit, for example, this post-translational modification step, and I can revise it, and I can say, well, I want really more than one phosphorylation event on this gene. Let's pick 2, right? And let's see what happens. And so now my number of genes is reduced to be expected, but again, it's not 0. And so now you have some things that may be of interest that you want to explore a bit further. And you don't necessarily have to stay within the proteomics world, right? So right now we did a quantitative proteomic expression experiment. We have a post-translational experiment here. I can add a step and I can ask, well, are any of these genes also upregulated based on RNA-seq evidence, right? And so I can pick an experiment here. And again, from any of these experiments, let's pick this gametocyte transcriptome. The result is probably going to be 0 because remember I only have 8 genes in my previous step, but let's give it a try. It doesn't hurt. So I'm asking for things that are differentially regulated, let's say in female and male gametocytes, and I can run this just by the defaults. And again, just demonstrating this. And so actually it turns out, I mean, I was very lenient in my search here in my RNA sequence search, but 5 of my genes that have at least 2 phosphorylation sites that are found both in the computationally and experimental apicoplast targeting experiments are also differentially regulated in female and male gametocytes. Okay, so let me go back to ToxoDB quickly. And remember, I ran this mitochondrial experiment. So again, just like within PlasmoDB, I can add a step and I can ask for post-translational modifications, and I can see what's available here. And you'll see that there are a couple of other types of modifications like acetylation, ubiquitin binding site, or monomethyl arginine modification. So I can pick any of these. Again, let's pick ubiquitin. I'm gonna add this. And again, I don't know what the answer is. The answer could be 0, but that's again, the cool thing about these in silico experiments is that you can actually run them and see what happens. So it turns out I have these 161 genes that are mitochondrial, are also potentially have ubiquitin binding sites, right? And then you can go ahead and take a look at them, and those may be of interest to experiment with a little bit more. Okay, so I think this was towards the end. I'm taking a look at some of the questions. If there are any questions
there's one
here.
that Eve is working on answering, but it's
Oh,
when
I
will
see
there
it
be
from
more proteomics data for post-translational modifications, and how would they contribute theirs, I think is what they're asking also.
Yeah, so this, I see the question is from Dinesh in Delhi. Welcome, Dinesh. I'm glad you're on the call. We constantly, scour the literature and hear from people about different proteomic experiments. We have a number on the schedule. Most likely, would have to look, but definitely, if you know of any experiments out there of post-translational modification, or if you have any, let us know. And the best way to do this is to click on Contact Us and just let us know about the experiment. And if it's published, just include the PubMed ID. If you have the PRIDE record for the repository, if it's deposited, just post that link. But the main thing is to start the conversation. And once you start the conversation, then that's when we can actually start working with you on incorporating the data. Depending on the data, sometimes we have to go back into the community and find out, if we have to prioritize things, we try and get input from the community. And soon you will be getting emails. We're going to open up some sort of community-wide community forums to hear about what are some key things that people need. And so hopefully we will start getting more feedback like that as well. But definitely, if you know of an experiment, let us know, and we will be happy to get it into pipeline. Okay, we'll give people a few seconds to ask any additional questions. While we are waiting, I'm going to pop up this. We have a post-webinar survey that helps us in, ask you questions like what else would you like to learn about, and that's important for us so we can actually better serve you. And of course, let us know how we did. You can obviously scan this QR code or type this URL. I'll put it in the chat window as well. So definitely start the survey. It's not many questions. It doesn't take a long time to do. And if you don't start it now, we know from past experience that you will not, most likely you will not answer the survey. So if you do start it now, it takes maybe less than 5 minutes to complete the survey. And your input is obviously highly appreciated. And as I mentioned before, this webinar is recorded and we will be posting it on YouTube. You will get an email tomorrow, both with a link to the YouTube tutorial or recording, and also a certificate automatically generated if you need that. In addition, the link to the webinar will be posted in our webinar page. So if you go to the webinar page, which was linked in the chat already, you'll notice here all of our previous webinars are listed here, and we will there'll be another line here for today's webinar, which will include a link to the YouTube tutorial. All right, are there any other questions or comments from people? Doesn't look like it.
Looks like it.
All right, well, thanks everybody for joining this webinar. Remember, next week we have a webinar that will cover HostDB. So if you're interested in host response to pathogen infection, we will be looking at various experiments in HostDB and how you can query them. And then the week after that, we're going to be going deep into building complex search strategies. So just like the search strategies that we were building today, which involve a few steps here and there, we're going to try and take you through some multi-step strategies that will leverage some of the cool features, including orthology, but also the phyletic pattern. So if you're interested, for example, in genes that are only present in fungi and not present in mammals, for example, because you're interested in finding things that are potentially nice vaccine candidates, right? And so we'll go through examples like that. We'll integrate in that various types of omics experiments, so proteomics, transcriptomics, and we'll build some really complex strategies that look complex, but they're really, step by step. They're quite straightforward, and we'll describe some of the cool features that are available to you when you build a search strategy. What you can do with that search strategy, how you can share it with others, how you can save it in your workspace, so it's available to you like a lab book, basically for future access. All right, with that, thank you, everybody, and we will see you next week, hopefully. Take care.
Bye-bye.
Bye-bye. Thanks, Suzanne.